So growing evidence suggests that normal brain development is impaired in neuropsychiatric disorders such as schizophrenia long before the onset of the first clinical symptom. As a developmental neurobiologist, a major goal of our research is to understand how the brain wires up during development and how this wiring process may go awry in neurodevelopmental disorders such as autism and schizophrenia and others. Now, animal models have provided a lot of important insight into these questions because they allow us to manipulate candidate genes and cells in a way that we can explore mechanisms. And this is something that's obviously very difficult to do in the human brain. So using mouse models a number of years ago, when I was still a postdoctoral fellow at Stanford, we unexpectedly identified a role for a group of immune-related molecules called complement. These are molecules traditionally associated with the innate immune system. And what we unexpectedly found was that these molecules were actually expressed in neurons and other cells in the healthy brain. So these were molecules that were there, and they were expressed particularly high during development when the brain was wiring up. And so today I want to tell you about our work and our discovery and where that's taking us, especially in the context of disease. Now, the wiring diagram of the brain is remarkably precise. So the human brain contains 100 billion neurons that make trillions of synaptic connections with other neurons. In fact, a single neuron is estimated to make thousands of synapses. And amazingly, these synaptic connections happen with the right partner at the right time in the right place. It's really mind-boggling, actually. So this is a picture. I don't know if we could dim the lights a little bit. This is an image of, in this case, a mouse brain that was genetically engineered to express fluorescent proteins in different subsets of neurons in the brain. And what you probably can't appreciate here is how interconnected these neurons are. They're of many different types, and they're actually, if we could dim the lights, that would be great. Then you can see down here this connectivity, which looks a lot like it's not precise, but in fact, it's incredibly precise. These neurons connect in very precise circuits. However, it doesn't start out so precise. So very early in development, the developing brain is incredibly plastic. It undergoes tremendous remodeling of these synaptic connections. So in a mouse, this happens over the first couple of weeks of development, and in a human, this happens over many, many years. And it's this refinement or sort of sculpting process, which is why a child is able to learn so many languages, and why a child's brain is able to pick up many, many new facts, many more than we can, at least at my age. So this plasticity is in part due to the fact that there is a lot of dynamic changes going on. So synaptic connections between neurons are forming and breaking all the time, and this plasticity is incredibly important. Now, what you can hopefully appreciate from this image is when you look at a human brain, and this was actually a really elegant study done by Huttenlocher back in the 70s, he actually stained autopsy tissue, human brain, with a special stain called Golgi stain, which labels neurons in a way that you can see their connections. And what this showed quite elegantly is at birth, you can see that the neuronal connections are quite sparse. And over the first several years of life in a human, you can see many more connections forming between different neurons, and this happens to be in the cortex of the human brain. But then what I really want you to pay attention to is what happens between the year of about 6 years and 14 years, where there's actually a drop in this connectivity. There's a loss of connections. And this loss of connections is called synaptic pruning, or synapse elimination. And this pruning process is critical for precise brain wiring. Just as important as it is to make synapses, it's also really important to break synapses. So this is a normal developmental process. And this process involves the permanent loss of some connections, but the strengthening and the maintenance of others. And a major question in the field is what determines which synapses stay and which synapses go. And that's one of the questions that my lab is really interested in understanding. And it's important in respect to disease because it's thought that a failure to prune synapses at the right time and place could underlie a lot of these neurodevelopmental disorders and neuropsychiatric disorders such as autism and schizophrenia. So, in fact, there has been emerging evidence that synapse loss does occur in the schizophrenia brain. This is just a real classic paper that was done by David Lewis a number of years ago, where he used that same stain, that Golgi stain, to stain autopsy tissue of the frontal cortex of schizophrenia patients versus normal healthy controls. And what hopefully you can appreciate is sort of a sparsity of these bumps called synaptic spines. These synaptic spines are depicted over here in more modern technique, where we can label a neuron with a fluorescent indicator. And all of those little bumps or spines, when you were to zoom in on one of them, what that represents is actually a synapse, right, between the axon of one neuron and the dendrite of another. So that's an excitatory synapse. And you can see this neuron has thousands of them, right? So in this experiment, what you can see is that there was a decrease or a loss of connections. And not everywhere, but he found them specifically in the frontal cortex and other cortical regions of these patients. So this was led to an intriguing hypothesis proposed over 40 years ago, the pruning hypothesis, that perhaps what's going on, and one of the mechanisms underlying the pathobiology of schizophrenia is synaptic pruning alterations. Now, this has not been directly tested, but this has been an intriguing hypothesis. Now, there's also evidence that synapse loss happens in other disorders, including autism. This is an example from a Rett patient. Rett syndrome is an autism spectrum disorder. And this was shown by Hoduzogby a number of years ago, that in human and in addition in mouse models of autism, you can see again a decrease in these synaptic connections. Not only is this true in neurodevelopmental disorders, but I also want to mention that in the adult brain, synapse loss is a hallmark of almost every neurodegenerative disease. The most prominent example is Alzheimer's disease, which we're going to come to later in my talk. And this is another example in an Alzheimer mouse model where you can see, again, a sparsity of these synapses onto those neurons in this model of Alzheimer's, but that's also been shown in the human brain. So the major question in the field, and one that my lab is very much interested in understanding, is how are synapses eliminated in health and disease? What are the basic mechanisms by which these synaptic connections are lost? And more specifically, you know, it's not just any synapses lost. We really want to understand what determines which synapse goes and which synapse stays. Are there molecules that determine early in development that this green synapse is going to be eliminated, but the red one will be maintained and strengthened? And so that's the question that we're really interested in understanding, because if one could understand the molecular pathways that regulate this process, it might lead to new insight and even therapeutic ideas and targets that could potentially protect synapses in disease. Now, for a very long time in the field, I think much of the focus has been on trying to understand neuronal mechanisms of synapse loss, right? And after all, synapses are between two neurons, right? This is a cartoon, a much oversimplified cartoon of a neuron. This is the cell body of a neuron, and all of these are their processes and dendrites, and these are the synapses from another axon, another neuron that's synapsing onto that cell. Now, we know, of course, that synapses are neuronal, but now this is sort of a neurocentric perspective, because what we're now appreciating is that neurons only represent half the brain. There's another group of cells in the brain that are not neurons. They're called glia. These are non-neuronal cells, and they actually represent even more than half the cells in the brain. And until very recently, neuroscientists have largely ignored them. They pretty much thought of glia as glue, which is where their name came from after all. They were support cells. They were the glue that held neurons together. But recent data from many labs over the last decade or more are starting to appreciate that glial cells play a very active role and a very direct role in regulating neuronal function, and in particular synaptic function and synapse development. And so what's missing in this cartoon is all the glia. In fact, you can't see them, they're not labeled, but they're everywhere. And they're very intimately associated with synapses and neurons throughout the brain. So I just want to kind of give you a picture of what we're talking about here. So this is just a picture now of showing lots of different cell types in the brain. Up until now, we've been focusing on the neurons, the gray cells. But in amongst all those neurons are these other cells. These are the glial cells, oligodendrocytes, and for example, these are microglia, which we're going to talk about in a moment. So just a cartoon version of this. Here is the synapse between two neurons, and there are multiple glial cell types in the brain. The main ones are astrocytes. They're probably the most numerous. And what those cells do is they intimately send out processes that touch those synapses, as well as blood vessels. You've got oligodendrocytes. They're the myelinating glia of the brain. They're the ones that wrap long membrane of the cytoplasm around axons and help to insulate or ensheath axons. These are important for increasing synaptic connectivity and conduction velocity. And there's another cell type, this one that doesn't look particularly beautiful in this particular picture, called microglia. Now, as a glial biologist and a developmental neurobiologist, I've actually spent much of my career studying the other half of the brain, the glia. Partly because no one was studying them, so I thought that was really needed, but also because there was so much to learn, right? There was so much mystery surrounding the function of glia and the brain. And I became obsessed with these cells as a graduate student. And I've actually marched my way through every single glial cell type in the brain, studying them in different contexts. But I've ignored one cell type my entire career until about 5 years ago, and that was this cell, the microglia. Why? Well, for one thing, there was very little known about their normal role in the healthy brain. Much of what we knew about microglia, these are resident immune cells in our brain. Much of what we knew was in the context of injury and disease. The other reason that I wasn't as interested in microglia, because I was a neurobiologist that studied development, was that we didn't think they actually, they're not born in the brain. They're the only glial cell type that's not born in the brain. They're immune-derived, meaning they come from monocyte-derived lineage. They actually now, we realize, through fate mapping studies, that these cells actually enter the brain extremely early in development, embryonically. And they actually sneak into the brain very early. They come from the yolk sac, and they migrate in, and they take residence in the brain. And they're actually there before any of the other glial cell types. And until about 5 years ago, we didn't even know that. Now that we know that, this opens up tons of questions. It leads us to wonder, what are those cells doing normally in the healthy, developing brain? But before we can ask that question, we better first think about what we know about them. Because actually, we do know a lot about these cells in the context of injury and disease. Now, I'm going to show you some movies, because I can't really articulate what microglia do without showing you in real time, okay? So this is using mice. Again, this is the power of mice, is that you can genetically engineer mice by putting a fluorescent reporter, GFP, or green fluorescent protein, into specific cell types in the brain. So in this mouse, this GFP was engineered so that all the microglia in this mouse brain are very conveniently labeled green. So when you look under a microscope, the only cells you see are those cells. And you can see they're quite beautiful. You can also appreciate that they tile the brain in a very interesting way. They only represent about 7% of our brains, right? Mouse brain and I think human brain. But the thing that you're probably not appreciating from this static image is that they're special and they're different from any other cell type in the brain because they're incredibly mobile. Okay, not their cell bodies, but their processes. And I want to show you what I mean. So if you were to, for example, and this is what we do in the lab, we can take this green mouse and we can put it under a microscope, believe it or not, and we can just make a little window into its skull, just thin enough that you can put a microscope on top of it and put fluorescent light through there and actually be able to visualize these cells. And then you can do this over the course of real time, over the course of minutes or even hours. And you can watch what these cells are doing. And so this is, again, a healthy brain of a mouse. And what I hope now you can appreciate is the fact that these cells are constantly surveying their environment all the time. And they're not resting at all. They're always moving their processes in and out, moving around, dynamically sensing the environment. And much like immune cells, their counterpart in the immune system are macrophages. That's what immune cells do. They're constantly surveying, right? So they do this, but they have these beautiful fine processes. The other thing that they do that's unique and different from most other cells in the brain is they have the capacity, again, based on their immune nature, to rapidly respond to danger and injury signals and local damage. So just an example of this. In that same experiment, if we were to make a very small laser injury into that mouse brain, very small, with a laser, what now you're going to hopefully be able to appreciate is what those microglia do. So this is the same, basically the same image field as I showed you previously, but this is after injury. So let's see if I can make this happen. Okay. So the injury is going to be right here, and look what they do. Crazy, right? I mean, if you don't remember anything else from today's talk, remember that. I mean, it's really amazing, right? So they did this very rapidly, and you could notice from that movie, and I will play it one more time because it's just so cool. And I wish Steve Hyman were here because he always wants me to play this movie over and over again, and I've got a fluorescent version of it now. So again, they move their processes. But what you can probably notice here is their cell bodies aren't really moving. They're just extending the processes. And what's later been shown is, obviously you probably are wondering what the signal is, right? So one of the signals that's been identified from Wenbiao Gan's lab a number of years ago is that ATP. ATP is what they're sensing. They actually have purinergic ATP receptors, and that's a damage signal, and they're actually chemotacting towards it. If you block the ATP, that doesn't happen anymore. So they can do this. So you can imagine in the context of a brain injury or traumatic brain injury, that's what microglia might be doing in the brain, which is both good and bad. So they're probably going there to protect, but they can also do other things like release neuroinflammatory molecules and cause more damage. The other thing that microglia do that's unique from any other cell, well, for the most part, they're also known as our resident phagocyte, right? So these are cells that can phagocytose or engulf things, eat things, right? So in the periphery, macrophages, they're the Pac-Man of our immune system, right? They are the ones that can rapidly engulf or eat pathogens or debris. Well, in the brain, the microglia also have that capacity. And this is an example, again, a live imaging movie of the cerebellum by my colleague Axel Nimron. And what this movie shows is we've made a very local lesion to that neuron in the cerebellum. And what I hope you can appreciate, and I'm going to play it one more time, is that the microglia that are associated with that one damaged neuron start to pull up the red material into their bellies, right? They're actually eating it. And it goes into their lysosomes. So they're engulfing all of that dead cells and all that debris. Now that serves a couple good things and bad things. It actually helps to clear debris from the brain and remove toxic materials like amyloid beta in the context of Alzheimer's disease. But it's one of their roles. It's their normal role, and they're in a normal condition, more of this sort of homeostatic state. But if you push them, or if you have a damage, they can do that. So the big question that we were interested in, and what was much less appreciated until very recently, was what do they do in the normal, healthy brain? And what is their function? And what I hope you can appreciate from this image was what I left out of the picture in the previous movie was the neurons. And now what I hope you can see is here is a process of one neuron. These are all those spines again. And what you can see is one of the things that microglia are touching are the synapses, are the spines. They're moving around and they're dynamically touching and surveying the synapses. And so this is just an image going into the visual cortex of a mouse looking right about here. And that's just to show you what they look like. And I think I have a movie of what that looks like zoomed in. And so here I'm going to show you a movie of microglia, again, dynamically touching these spines all the time. So they're doing this all the time. We think this is a homeostatic process. And we and others have even shown that they can sense neuronal activity and neurotransmitter release and signals that the neurons are releasing. So they're well poised to be communicating with neurons and responding to signals from neurons. And so this led us, a number of years ago when I started my lab, to ask the question, what are microglia doing in the normal developing brain? And could they be helping to modulate and prune those developing synapses, given that they're phagocytes, they're dynamically interacting with synapses, and they have immune-like functions? Could they actually be in a normal brain plucking off and eating those extra synapses in the brain? And that's the hypothesis we set out to test a number of years ago. To test that hypothesis, though, we needed a model system. And so we turned to the mouse visual system as our model because this was a really beautiful model that neurobiologists have used for many years to study synaptic remodeling. And I just want to take you through the model very quickly. So this is just the mouse brain, a cartoon, showing the eyes projecting, if you were to put a dye, a tracer, in this case a blue and a red one, into the two eyes of a mouse and follow where their connections or their axons go, you can see they make two connections. One in the thalamus, the visual thalamus, called the DLGN, of the dorsolateral geniculate nucleus of the thalamus. So this is your visual relay station en route to the visual cortex. So you can actually follow their projections and ask where they go. Now, I've switched colors on you, unfortunately, here, but what I'm showing you now is a cross-section. If we were just to cross-section our way through this, This is now a cross-section of the same thing, showing that the axons from the neurons in the eye, the retinal ganglion cells, send these long axons that project to very specific targets and neurons in the visual thalamus here and all the way up to the visual cortex. And they actually form these eye-specific territories. And that's part of how we have binocular vision and specific vision, because the information from your left eye and the right eye come together in a very precise way so that you essentially have a map in the visual parts of your brain. And this is just a further cartoon showing, again, if we could put the dyes in the eye, you could see that they send these axons, and early in development there's a lot of intermingling between the axons. And then over the course of the first couple of weeks in development, they then segregate out into these eye-specific territories where there's very little intermingling. And this process represents the pruning of the inappropriate eye inputs and the strengthening and maintenance of the correct eye inputs. And here's a further cartoon. If you were to zoom in on a neuron, the perceiving side of this, you would see that a typical neuron early in development is binocularly innervated, so it's innervated by axons from left and right eye. But then a mature neuron is innervated from axons from either left or right eye, but not both. Again, this is an example of pruning. So given that pruning happens, we know when and where it happens, and that we have the ability to very conveniently label or trace the projections, Dory Schaefer in my lab decided to use this clever approach to test the hypothesis that microglia could be engulfing those inputs, right? So what she did is she put, again, dyes in the eyes of a mouse. She, again, used these transgenic mice in which all the microglia were green, and they're all hanging out in the LGN there. And then she simply asked the question, are they eating or pulling up bits of those synaptic inputs or the endings of those axons during development? And if so, what's regulating it? And so instead of engulfing big bits of dying cells, there's no dying cells here. This is a healthy brain, and we want to know if they're plucking off and eating the little bits of the endings of those axons, which are shown here in red and blue. And what Dory found is indeed, here's an example of one microglia. All of those puncta, those little blue and red puncta, are the endings of the axons, and we're going to remove everything that's not within the microglia, and we can do fancy 3D reconstructions and surface rendering. And when we did that, we realized that every microglia that we surveyed during the peak of pruning in the mouse brain had many, many inputs, synaptic inputs, the terminals inside their lysosomes, inside their bellies. And in fact, what was really quite striking is that this was really precisely happening in this window of development. If you look later, like a month-old mouse, when pruning's done, no more inputs within their bellies anymore. So it's tightly regulated. It happens at the right time in the right place. And this was the first evidence that microglia are a cell that's participating in this pruning process. And this, of course, led to the big question, how are they doing this? What are the molecules? What are the mechanisms that tell microglia which synapse to go for, which synapse to engulf? How does that work? And so we started thinking a lot about this. And again, we constantly turned back to the immune system for clues, given that these cells are immune-like, and they have a lot of receptors and pathways that are much like their counterparts in the peripheral nervous system, the macrophages. So they are different from those cells, but they share a lot of common features. And so we started thinking about one particular family of proteins called complement. And the reason why, you'll be more clear in a moment, but one of the reasons why we're interested in complement is that it is a classic eat-me signal in the immune system. It is how macrophages in your immune system recognize and clear unwanted pathogens or debris from your body. So the innate immune system, this is our first line of defense, the complement system for removing pathogens. So before your slower adaptive immune system, it's the complement system that can act very quickly. And it's a group of secreted proteins that are in your circulation. And when encountered, let's say a bacterial cell, C1q, the initiating protein of the cascade, binds to that or tags it. That initiates a proteolytic cascade leading to the binding of the C3, which is a very strong tag or opsin. And any cell that has C3 on it gets phagocytosed by microglia that have complement receptors. And so this is one of the main ways that complement tagged material gets removed from your body. So we wondered, could complement be similarly tagging synapses for elimination, much like they would tag a pathogen for removal in the immune system? And indeed, we found and published a number of years ago that complement proteins These are proteins expressed in the brain normally during development, as I told you at the beginning. So we were surprised to find that a number of years ago. And that if you stain the brains of mice and humans, for that matter, we see C1q and C3 targeting subsets of synapses in the developing brain. So they're actually there on synapses, not all synapses, and that microglia are the only cell in the brain, in the healthy brain, that express receptors for complement. So putting all this together, this led us to hypothesize that perhaps complements were tagging subsets of these excess synapses during development, and that one of the ways they get removed is by these microglia that recognize it and then initiate the removal. And indeed, we went on, we showed a number of years ago that mice that lack, these are genetically engineered mice that lack C1q or C3, right, the two components in the pathway, mice that don't have C1q or C3 fail to prune properly. They don't segregate into these eye-specific territories, and their neurons remain multiply innervated. So we knew previously that these molecules were necessary for pruning. This was a big surprise a number of years ago, but we did not have any idea how this was working. And so that's what we then started putting the two stories together. And the hypothesis is that synapses, or subsets of these developing synapses, are bound to complement proteins, and that microglia that have receptors for complement recognize and initiate that engulfment. So to test that idea, we actually took advantage of the fact, again, a genetically engineered mouse that lacks this receptor. So this is the only cell in the brain that doesn't have the receptor. So no longer, even if complement's everywhere, it can't recognize it anymore. And we ask whether mice that don't have complement receptors, do they still engulf synapses and do they still prune normally? And what we found and published was that indeed mice that lack this receptor, they're only about half as good at eating synapses, and that in fact they do have pruning defects, and one of the outcomes or the functional consequences for not having this mechanism in place during development is there are too many synapses in the brain in this case, right? So they're not pruning. They're hyper-connected, hyper-wired. And a colleague of ours went on to show, David Prince, that mice that don't have, that have too many synapses and that lack complement actually are more prone to seizures. So they actually have seizures because they have too many synaptic connections. So that's one of the consequences of not pruning properly, is you have a hyper-wired brain. And so we're now, of course, looking into running a lot of behaviors on these mice to better understand how, what the functional consequences are in behavior when you don't prune properly. So pulling all this together, what I hope to be able to sort of tie together here is what I've told you is we've identified one immune-related pathway that's important for pruning. And by no means is this the only mechanism of pruning. There's many other mechanisms, some of them neuronally regulated. And interestingly, a number of other labs over the years have gone on to show that there are actually a number of other immune molecules that are important in pruning. Carla Schatz, long before our paper, actually showed that MHC class I molecules, these are members that are proteins traditionally associated with the adaptive immune system, were also necessary for pruning. And more research is emerging that implicate these immune molecules in pruning in the brain. And again, these are not in the context of infection or disease. These are molecules and genes that are expressed by neurons and glial cells normally, and they have a different role in the brain from the immune system. They share some common features, but they actually have an unexpected role in brain development and brain wiring. Now, I told you that when you lack this pathway, you have too many synapses. But one idea, or one hypothesis, is what happens if this pathway was overactivated, so too much pruning, too much complement, too much microglia synapse eating. We hypothesized that this could be underlying some of the synapse loss in some of those diseases that we talked about. And indeed, just going back to the beginning where we started here, could some of these mechanisms that I just described to you today, could they be aberrantly regulated in such a way that they could be one of the contributors to synapse loss in these different brain disorders? And this is a hypothesis my lab is currently testing. And I'm going to share with you some unpublished work, that just a few slides now, where I'm going to share with you some of the work we've been doing in the context of Alzheimer's disease. And we chose Alzheimer's disease first because this is a disease that's obviously a devastating neurodegenerative disease, but there's quite a lot of evidence, both in human studies and in mouse models, that synapse loss does occur, and it occurs very early. And because there are really outstanding Alzheimer's mouse models, and a lot of them, we had the ability to start to test this hypothesis in a very clear way, which I think is a bit more challenging in disorders like schizophrenia and autism, where we don't know exactly where and when to look. So I want to take you through some of this recent data. And just to sort of set the picture, as many of you know, Alzheimer's disease is a progressive neurological disorder, probably the most prominent and common form of dementia in the age population. It was first, this is the first patient ever to, so Dr. Alzheimer's actually first characterized it in this patient, Auguste Dieter, who actually came in with these really striking memory and cognitive impairments. And later, when they looked at his brain, it was the first brain, an Alzheimer brain, that was analyzed and looked at in terms of its pathological symptoms and hallmarks. And what you can certainly appreciate here is the decrease in the size of the brain or the cortex in an advanced Alzheimer's brain. And two of the hallmarks that many of you probably know about are Alzheimer or A-beta plaques and these neurofibrillar tangles that occur in the brains. These are the pathological hallmarks of Alzheimer's disease. But I want to just make a point here, because there's a lot of folks, of course, working on Alzheimer's and trying to understand how this works and how to slow the progression and ultimately to treat Alzheimer's disease. But one of the things that I want to point out is, again, that synapse loss is a much stronger correlate of cognitive decline and memory impairment than those pathological hallmarks I just told you about. And the other thing I wanted to tell you is that there's a lot of evidence now that synapse loss and dysfunction happens much earlier than those hallmarks, than those pathological hallmarks, and of the cognitive decline. So this is one of the earliest events, and it's thought that it happens years before you actually show symptoms. And in mouse models, that's also been shown to be true. And so given that, we wanted to know, using mouse models, whether complement and microglia-mediated pruning could be underlying some of this early synapse loss, and to test that hypothesis. Now, there is actually quite a lot of evidence to implicate both microglia and complement in Alzheimer's disease. So there's a lot of work on this. And this is just an example to show the A-beta plaque is here in the middle, and it's not stained here, but what I'm showing you here, all these cells around it are the microglia. And no, I don't have any live movies here, but they surround these A-beta plaques. So they're known to play an important role both in engulfing or phagocytosing or clearing the plaques. So this is actually a good function of microglia. But they're also known to be powerful mediators of neuroinflammation. So they release a lot of cytokines and proteins that are important to mediate neuroinflammation. So they have both good and bad roles in Alzheimer's disease. But much of the emphasis on these cells in Alzheimer's has been late-stage Alzheimer's. Similarly, complement proteins are dramatically upregulated in human Alzheimer brain. There's been quite a lot of work on this as well. This is just an example of C1q and C3. If you were to immunostain the brains of these individuals, you could see that there's a lot of deposition of complement molecules, again, especially around those plaques. So complement is very high in Alzheimer's brain and in mouse models. Microglia are very activated. They surround plaques. We know that. But the big question we wanted to address in the lab is, could they be playing an earlier role? Could they be, are they a cause or a consequence? And that's been very difficult to tease out. And so there's actually been also a number of different genetic studies, GWAS data, linking and both complement and microglia to Alzheimer's disease. So two genes in particular, CR1 and TREM2, this is a microglia-specific gene, increased risk of Alzheimer's, but mechanisms unknown. And there's a lot of people trying to understand what that means and how that's working, and how these genes are increasing risk of Alzheimer's disease. So putting all this together, we asked the question in my lab, what are the mechanisms that drive early synapse loss in AD? And ultimately, we want to know, could we prevent or slow synapse loss and ultimately cognitive decline? We're looking for mechanisms. So we hypothesize that early synapse loss is a reactivation of this developmental pruning mechanism. And one of the pathways that we're studying is, of course, complement and microglia. And so my postdoc, So-Yan Hong, in collaboration with Dennis Selkow at Harvard Medical School and Brigham and Women's Hospital, we've been embarked on a collaboration a number of years ago to test this hypothesis using Alzheimer's mouse models. And we want to know whether we have a reactivation of this pathway in the Alzheimer brain. And so we specifically, Soyeon wants to know whether they are engulfing or aberrantly pruning synapses in Alzheimer's models. And this is just some unpublished data to show that using the same kinds of techniques we used in development to label the synapses as well as the microglia, Soyeon came up with a way to do the same kind of engulfment analysis. So are they actually engulfing synapses? And what she showed is in these Alzheimer's models, there was significantly more engulfment of synapses early in Alzheimer's mouse models. And the other thing that we've been interested in is whether complement is mediating this process. And we've actually been staining some Alzheimer's mouse brains with complement. And of course, we knew that complement was high and it was localized to plaques. But now we also have evidence that complement, in this case, it's an immunostaining with an antibody that recognizes C1q. We see C1q, and this is in green here, targeting subsets of synapses in the Alzheimer's brain. And so this, and we also see this in our mouse model, and we see it quite early, before you start to get cognitive dysfunction and memory loss. So the question is, does it mediate or contribute to the synapse loss in Alzheimer's disease? And so to test that, what we can do is we can take Alzheimer's mouse models that have been genetically engineered, and we can cross that to a mouse that lacks complement molecules, for example, C3. And so by crossing a mouse, these two mice together, you can make a mouse that has Alzheimer's but lacks complement and compare that to its control littermates that have complement. And we can then measure synapse loss and disease progression. And this is just an example. What we do is we quantify the number of synapses in these mice. We stain the brains with these synaptic markers. This is a marker that labels the presynaptic axon, and this PSD95 labels the postsynaptic. And so by doing that, we can quantify the number of synapses. This is what we're actually looking at here. All those puncta, if you see a yellow puncta, it's when those two synapses come together. And what we find is that there's a striking decrease in synapses in the Alzheimer's mouse models. But in mice that lack C3, we see protection against that synapse loss. So this is exciting to us because it suggests that a pathway that we know a lot about, a pathway that mediates normal pruning, may in fact, if manipulated, in this case genetically, but of course ultimately we're trying to use inhibitors and small molecules and therapeutics to be able to go in there and see if we can block complement and rescue the synapse loss. And we have preliminary data that also suggests that we also can rescue some of the cognitive impairment and some of the context-dependent memory loss that these mice actually have. And so taken together, we're very much interested in expanding this work into other disease models. And here, as a member of the Stanley Center in the Broad, in collaboration with Steve McCarroll and others, we're very interested ultimately in asking the very big question of whether our pruning mechanisms that we've described and discovered, could it actually be one of the mechanisms underlying the pathobiology of schizophrenia and the synapse loss that's been shown? And so there's actually some emerging genetic data from the Broad and from others now that are implicating molecules in the complement cascade. So stay tuned, but we're very excited about this. And this has begun the beginning of a really beautiful and exciting collaboration between my lab, Steve McCarroll's lab, and others. And so we want to be able to design experiments to test the pruning hypothesis in schizophrenia. And ultimately, we hope that the work that we're doing on my lab on the basic level, basic science, can provide novel insight into therapeutics that might be able to slow the disease progression in schizophrenia and other neuropsychiatric and neurodevelopmental disorders. So I'm just going to end by thanking a lot of people, just my lab and a lot of incredible collaborators and funding, and all of you. So thank you very much.
Thank you, Beth. That was great. So we'll go ahead and take questions. You can either line up at the microphones on either side, or if you want to tweet your questions to #BroadTalks.
Beth, Manoli Scalis from MIT. Wonderful, wonderful talk. So this whole use it or lose it aspect of brain function, there's a very simple, naive idea from watching your absolutely beautiful data that's, where the complement would basically go to the synapses that are actually not being used.
Right.
Where the neuronal connections somehow in the firing would actually remove complement, and if you don't fire, complement would accumulate. Have you made that link?
Yes, we have. So you're exactly right. The idea of use it or lose it, the synapses that are going to be lost in your brain are the ones that are less active at the expense of the ones that are able to fire that postsynaptic cell. And we have data now, both published and unpublished data, that suggests that the synapses that get tagged are the weak ones. And we've actually been able to use optogenetics and this DREAD system to be able to genetically manipulate activity in such a way that we can drive competition. And when we do that, we can see a selective tagging of the weak synapse. And we have data published that when microglia are pruning, if we block activity in one eye with TTX, they preferentially engulf the less active or silent synapses. So putting together those two pieces of data, we hypothesize that this is a way that they're targeting the less active synapses during development.
Absolutely fabulous. And would that be a slow process of accumulation of complement that gets kicked out by activity, or what's the actual mechanism by
so
which
we're time-lapsing this now, and we're trying to get a better sense of, because these are secreted molecules, and what I didn't tell you yet is, and a question that we're still trying to understand is, what is it binding to on the synapse, right? So these are secreted molecules, but yet there's a specificity. So that suggests there's some molecular receptor on certain synapses, i.e. the weak ones, that's causing complement to bind to it. So one of the projects in the lab now is to try to fish out and identify what those molecules are, because if we could understand that, that could provide a lot of insight in terms of therapeutics as well.
Phenomenal work. Thank you very much.
Thank you.
Hello, my name is Ben. With regard to your research, you're looking at trying to slow down the development of a loss in synapses. Is there any future, if you look further into the future, is there any evidence of regenerative medicine being able to develop new synapses or develop, we have brain loss, new growth itself, period?
Oh, it's a great question, and it's a really hard one, actually. So in general, I think there are certain parts of your brain, like the hippocampus and the olfactory system and other parts of the brain that are probably remodeling synapses all the time. So in development, that loss is permanent. But in adults, you can imagine that there's some plasticity there. And it's not yet clear, what is regulating the formation of new spines. But one can imagine a scenario where people have been studying spinogenesis, making new spines. If you could tap into those mechanisms, you might be able to find a way to add more. The caveat of that, however, is, as I started at the beginning, there's specificity here. So if you just started making more connections, it has potentially adverse effects in that you could have too many connections that are not the right connections. So I think you have two challenges. One is to, if you are going to try to promote synaptogenesis or synapse gain, make the right ones and make sure that the right connections that are going to be functional. And I think that will be much harder, although I'm sure there are folks that are working on that. So it's a great question.
Thank you.
So following up on Ben's question, would you anticipate that if you have subsequent synaptic formation or neurogenesis, adult neurogenesis, would you anticipate that you would have the same overwiring and then pruning back as you see initially, or?
Yeah, it's an excellent question. So my graduate student, Janelle Wallace, is actually testing the hypothesis in parts of the brain that undergo adult neurogenesis. So, as many of you probably know, there are a population of neurons in certain parts of your brain that are constantly turning over new neurons. And those new neurons integrate into circuits, and they form functional connections. One of the questions we want to know is, do they go through a similar refinement or pruning process to make sure when they get in there, they're connecting to the right partner? I can't imagine that wouldn't happen, but it's not yet clear whether it's the same types of mechanisms or something different. So Janelle is actually testing that hypothesis in the olfactory system, where you can actually watch it, using time-lapse imaging, watch these newborn neurons integrating into circuit in real time, and then asking whether they undergo remodeling. And then if so, we can start to interrogate mechanisms. But that hasn't been done yet, but I think it's a great question.
In that telling image of the microglia responding to the injury in the mouse, what's the time scale on that? Does the microglia cascade in response? And how is there any way to find out what is responding and why? Is it like job assignment or that
right?
Sort of thing?
Yeah, it's seconds. It's very rapid. That was almost real time. I mean,
Okay.
it's incredibly fast. Their processes are just always surveying all the time. And again, it seems to be more of their distal and their fine processes, not their whole big processes. But if you push them, you can start to get really dramatic changes. And again, one of the mechanisms that was identified, not by our lab, but by Wenbei Gan and now others, is that ATP, a purinergic signaling molecule, is really important in that process. So it's quite striking because no other cell can do that. So they're recruited to the scene very rapidly, where they can potentially help the situation, but they can also cause damage and harm.
Yeah.
So there's a lot of interest in the field to better understand what's regulating their motility, and then when they get there, what are they doing there in terms of functionally? That's not really clear.
Can you identify the ones that aren't responding, what they're doing?
You can, again, by imaging them. You can follow those that are less motile versus those that are more motile. And now we and others are trying to develop, using these in vivo models and also in vitro models, you can start to add different antagonists or drugs or manipulate different candidate pathways to see what alters their ability to chemotack towards or towards a synapse or towards a cell. So one of the things that we're really interested in doing more specifically in the context of synapses is that is a very dramatic situation where they're going towards an injury. But we want to know, in the context of synaptic activity, if you were to have a neuron here that was firing much higher frequency than this neuron, and that would mean that that neuron might be releasing more neurotransmitter, more signals, could the microglia in the local environment know that? And it seems like there's evidence that that is in fact true, because you can manipulate activity and it changes their dynamics. What's the signal? And we don't know what that signal is yet. I think that's going to be really important.
Thank you very much.
You're welcome.
Thank you very much for the nice talk. I have a quick question. So you mentioned that there's several mechanisms for pruning the brain. And is there any knowledge why and how those mechanisms are different, for example, for microglia versus another mechanism?
So there are a number of other mechanisms that have been identified. Not as many molecules as I would like to see at this point, but there are mechanisms. And one of the mechanisms that's also is thought to be cell intrinsic. So this process of retraction or autophagy, right? A cell actually pulling back a part of its own membrane. And so there's some evidence that that's actually also going on, that some of these mechanisms could be controlled by the neurons themselves. So what I described today are extrinsic signals, secreted molecules and cells, right, that are doing this. But it's very likely that our pathways are interacting with these other pathways, because I think in the end, you need to have clearance of the cell. And what we don't know yet, which is actually going to be an important question to answer, is whether during pruning, something else in the neuron, even intrinsic in the neuron, initiates the pruning, meaning that there's a change or a retraction or a local change, and then the microglia are responding to that. Is it secondary, or are they coming along and initiating or plucking off an intact synapse? So without time lapse, that's been really hard to do. So what we're now setting up to do in the lab is, and Dory Schaefer was the postdoc who started that work and is now in her own lab, is using those same 2-photon imaging approaches, be able to watch this pruning happen over time and see if we can catch them in the act and see what they're doing, what comes first. So we don't yet know. But I would say that there's multiple mechanisms. And as I mentioned in my talk, that another group of immune molecules that were identified are these MHC class 1. These are molecules involved in the adaptive immune system. And Carla Schatz has a number of beautiful papers implicating this set of immune molecules. And what's so interesting is that the phenotypes or the consequences in the mice that lack her molecules, the MHC class 1 and complement, are incredibly similar. And there's some evidence that suggests there may be some interaction between the MHC and the complement. So I think another big question is, could these molecules be, in the end, working together in some way? And that's, I think, another big question that we're very interested in addressing.
Thank you.
I just wanted to interject with a question that was tweeted from the audience. Do you know what triggers the healthy pruning period and what stops it?
Yeah, it's a great question. So one of the things we know is important and has been known for a number of years, and it was mentioned earlier in a question, is neuronal activity is an important mediator of pruning. And so it's known that, spontaneous activity in the retina, for example, is one of the processes that sets up this pruning process. Again, the molecules linking activity to that have been mysterious, but it could be that one of the regulators upstream of our pathway could be neuronal activity. So I described one way that that might work, in that activity could set up a local competition between competing inputs, and the less active one, by some molecular change, is going to lead to the tagging of that synapse. But we have also some data that if you fire a neuron, if you make that neuron stimulated to fire action potentials, that can also turn on transcriptionally the upregulate C1q at the transcriptional level. So activity could play many roles, including expression of those molecules. And in fact, Carla Schatz has shown a number of years ago that MHC class 1 is also regulative activity. So you can imagine activity being a regulator of multiple different levels of this. And so that's my hunch in terms of what's upstream is activity. Yep. Great question.
Okay, this is a broader-based question
Yes.
around Alzheimer's.
Yes.
Is all of this, do you have any idea yet if all of this is informing some of the traditional insights or suspicions around Alzheimer's? I'm not an expert at all in this, but there's correlations with things like aluminum, zinc, et cetera. Is there anything that we're learning from this in terms of where to go in other areas of Alzheimer's yet, or is it too early?
It's a great question, and I think the one thing that I think, and this is, again, my opinion, but I think that, and this is based on a lot of emerging data, that the early synapse loss that's happening in Alzheimer's is happening long before you start to get, so this is a very early event. And a lot of the research as well as the therapies are not going early enough. And so my hope is that we can start to focus on the earlier time point. And the other thing to say is that there's probably multiple different things going on here, and that one thing's probably not going to solve it all. But if you could find a way to slow that synaptic loss, which is most correlated with the memory loss and the cognitive impairment more than anything else, that's what's so striking about Alzheimer's disease. We all know that, right? So if we can find a way to protect synapses, and there's probably multiple ways one could do that, I think there's some hope for slowing the progression of the cognitive decline in Alzheimer's disease. And since this is such a late adult onset disease, this could make a big difference. It could slow it by years. So that's where our focus is, and I think the focus of more and more labs. But there's a lot of camps in Alzheimer's, and there's a lot of people that are trying to get rid of the plaques, try to get rid of the A-beta, trying to fight inflammation. All of those are important, because once you have it, you want to also deal with that. So it's going to be a multi-pronged approach, I think. And the other point I want to make from this is that we have evidence, and there's evidence from other labs that is also emerging, that synapse loss is also happening in a lot of other neurodegenerative diseases. So Huntington's disease, frontal temporal dementia, Parkinson's disease. There's a common denominator among all those diseases. So even though each of those diseases has different genetics, right, they have some common features, and one of them is synapse loss. And so one idea is that there may be some common pathways that if you can figure that out, it might be relevant across the board. And that's our ultimate hope, is that that might be a better way to go at it. So thanks, great question.
So I think we'll have time for 3 more questions, 2 from over there and 1 from over there. So would you like to go ahead?
Thank you very much for that talk. Your data on Alzheimer's in C3 in mice is absolutely fascinating. I have 2 questions related to it. Does the persistence of synapses in the C3 knockout Alzheimer's model, does that match with a failure to reduce cognitive function?
Yeah, so we have data now, I didn't have time to show, and it's still unpublished, that when Cindy Lemire, our collaborator, did the behavioral analysis on those mice when they were older, so a 16-month-old Alzheimer mouse model, and ran them through a number of behavioral tests, those mice that lack complement, C3, did better on the context-dependent memory tests, in certain of them. So that suggests that not having complement there, which we know protects the synapses, also ultimately results in some improvement in some of these tasks. The interesting thing, however, is that those mice had more plaques. Okay, so that's really interesting, because there's other data suggesting that there's a disconnect between plaques and the cognitive aspects. So things that treat the synapse loss are probably going to be more helpful for the cognitive memory impairment, and things that treat the pathology, like the plaques, may be important for a lot of other things, but they may not be as correlated with the cognitive tests. So I think that's a really interesting observation. So we'll see. But I think ultimately we want to know, run these mice through multiple tests, do very extensive characterization to see what aspects of the disease progression is it helping or not.
One other question. How did the prevalence of synapses in the C3 knockout Alzheimer's mouse compare with the non-Alzheimer's C3 knockout?
In terms of from the beginning?
Yeah.
Well, interestingly, although the sensory systems, like the visual system, it's very important in normal developmental pruning. In the hippocampus, at least, they start off with the same number of synapses. So if you look in a 1-month-old mouse, in the Alzheimer mouse that does and doesn't have complement, their synapse numbers are roughly the same. And then it's as the disease progresses that it seems to be protective. So it's a way of potentially separating out development from disease. But the way to really do that is what is being done now, which is to conditionally manipulate complement later. So get rid of the developmental aspect of it. So making conditional knockouts where you can knock out complement when you want, where you want, in a more specific way. And that's still all ongoing work.
Thank you very much.
Yeah, you're welcome.
Hi, great talks.
Thank you.
So I guess I was really struck by this slide that showed this microglial clustering around the plaques in Alzheimer's brains. Is what that suggests that maybe the microglia and complement cascade is like somehow mistaking amyloid plaques for some sort of cellular injury leading to localized synaptic pruning? And
well, yeah.
Yeah, do we see evidence of increased pruning in those areas immediately surrounding them?
It's an excellent question. So there's a couple of things we know from the work of many in the field. Is one of the things that we know that microglia are doing around those plaques is they're recognizing things like complement that are sticking to Alzheimer's plaque, to A-beta plaques. So they're the eat me signal that's essentially saying, clear me, get rid of me. And remember, one of the things they do, the good things they do, is to clear debris and things like that and toxic things. So they're going in to help, to clear those plaques. But the other thing that we haven't yet explored, which is what's hard to tease out, is surrounding the plaques. We know from the work of Brad Hyman and Tara Spires that there's a lot of synapse loss that happens right on the periphery of those plaques. So that's what we're trying to do, is to kind of go over there and see if we can see them engulfing those plaques in the Alzheimer brains, engulfing the synapses around the plaques versus the plaques themselves. And so we can label the plaques one color, the synapses another color, and we can start to do those kinds of engulfment assays to address that question. But more than likely, the microglia being around the plaques have multiple roles. They can clear plaques. They can also, they're actually contributing again to the neuroinflammation that's going on, but they can also prune synapses. So we think they wear many hats. And I think the key is going to be to tease out their role. And I think one of the ways we want to do that is to do it early versus late. Because once you get late, there's so much going on that it's very hard to tease out specific functions. But if you rewind the clock to the beginning, you can start to ask, what are they doing? Are they mediating? Are they contributing? Or are they sort of responding? And I think that's kind of what we're trying to do.
Right. And do you think that it's just, okay, the microglia are trying to clear the plaques so there are more in the area, so naturally you would have more synaptic pruning, or is there some interaction specific to amyloid beta that has to do with this?
Yeah, there is, and I left it out because it was too much to say, but you are right on. And one of the things we know is that, so A-beta, right, is a small protein, and when it is in this fibrillar form, it's forming those plaques, right? However, in their monomeric form, or their oligomeric form, when they're secreted, what we know from the work of Dennis Selkoe, Bernardo Sabatini, and others, many others now, is that secreted form, that particular form that doesn't make the plaques, that's synaptotoxic. So if you sprinkle that or put that onto a brain slice, or if you give it to a mouse, it is mediating synapse loss. So that suggests that A-beta, which is what makes the plaques, it also has other roles, which could also be mediating the synapse loss. And so that's the thing. I think it's a great question, and we actually have evidence to suggest that it's C1q and A-beta working together in that model I showed you today to mediate the synapse loss. How that's working, we don't yet know, but there's some connection between A-beta and C1q, and that's sort of where we're headed.
Wow.
Yeah, it's crazy.
That's so
crazy
compelling.
That's
great.
Hi, thanks again for the talk. And for the last question, I'm going back to the first question and about this use it or lose it way of pruning. So the question really is like, so for a layperson like me, like looking at the cartoon of the visual cortex of the mouse, it seems there are obviously more connections, like cross connections on different sides of the brain than the direct connections on the same side. And am I correct in thinking that it starts with more connections on both sides and then connections are pruned? Or in general, are there specific structures which evolve from this pruning process? And if so, it's not just symmetry breaking in some evolution of statistical process, right? Because in that case, there could be more than one structure that you can end up with, which does the function. So are there other signals? Why are there more connections, like more cross connections for the visual system? Or if there are specific structures, how do those evolve?
Yeah, it's a great question. I don't have the answers to that question, but I will just put this in a bigger picture here. We focus on the visual system because it's a sensory system. And sensory systems, like your auditory system, your visual system, those systems prune pretty early in development in a very stereotyped way, kind of like what we talked about today. We know where it happens, we know when it happens, and it's how, it's essentially how, the information from the sensory system, the retina in this case, is sending the information to the brain, and that's helping to prune. The big question is, what about other parts of the brain that aren't sensory? And of course, there's evidence that different parts of the brain prune at different times, and maybe by different mechanisms. So for example, in the cortex, there's lots of connections between neurons from the same layer of the cortex. Are those connections being pruned? And if so, when? And one of the areas that we are really interested in trying to understand is the prefrontal cortex, because this is the area in schizophrenia and neuropsychiatric diseases that's actually where the synapse loss was observed. So we want to better understand where and when pruning happens there. And it's thought that that is happening quite later. Like in the human, that could happen when you're preteen, right, 12, 13 years old. We don't yet know in mouse models how that's working. It's really hard. We don't have convenient ways of tracing the processes and doing the kinds of things that we did today. But we're trying to use new technology and new imaging approaches to try to test and ask those questions that you just posed, which is pruning happening all over the brain? Are the same mechanisms regulating pruning everywhere? Or are there distinct mechanisms for each system or each circuit? And that's a question, I think. I think there are going to be some common features, but I think we really need to tackle that because disorders like autism and schizophrenia, it's involving multiple brain regions. And we need to better understand those circuits, and we need to understand how they're being developed and potentially pruned. And I think that's the big challenge that lies ahead.
Thanks.
Yeah.
Great. I want to thank you all for coming, and I wanted to thank Beth Stevens again for
Thank you all.
joining us tonight.